I'm standing right now next to the Great Bay Estuary. I'm at Adams Point, which is located in Durham, and it's a beautiful place to come and enjoy the bay. You may never have heard the word estuary before. Estuary is defined as the place where a river meets the sea. There are rivers around us that drain into the Great Bay. And then there's also salt water that comes in from the ocean, and that forms the bay. This bay was formed a long time ago, about 14,000 years ago, and now it is home to many animals, young and old. Some of those are in need of protection. There are 23 species that are on the threatened or endangered list. So they are in need of some kind of protection. And luckily, there's a team of scientists that work here to study the bay. And we're going to meet one of them today. Her name is Anna, and she is a graduate student from the University of New Hampshire. And she's very interested in the quality of the water and the nutrients in the water. So she is going to explain to us how she collects data or information at many different places, right by the rivers where they meet the bay, in the bay, and then where the water is leaving the bay. She takes that information and she analyzes it or interprets that data. And that's something that scientists do that I'm very excited to kind of talk about today and have you learn a little bit more about. It's something you can do too on any experiments or observations or investigations you're doing. You also can take that information and analyze it. So right now I'm going to stop and show you a map so you understand where Great Bay is. It's right near us, just a few miles from our home. And I'm going to show you an animation that acts out what's happening with the water and how the water empties into the bay. I'm going to show you some of the animals. And while you're watching, I would like you to think about and pay special attention to how Anna, as a scientist, works to understand a lot about the bay and how she analyzes and interprets that data or that information. And also I hope you can learn some really exciting things about the bay. It's just a place that's right near us, and it's so interesting to kind of learn some more about how it works as a system, as an ecosystem, and how maybe you might help to protect it. So I'll start with the map and show you a little bit more about Great Bay before we meet Anna. You probably heard the geese just now out on the estuary. There are many of them out on the water right now, and some have come down from Canada and will be spending the winter here on Great Bay. There are also beautiful bald eagles that nest here and depend on the estuary for their food. Great blue herons wade in the bay, striped bass swim in the water. You can look at the map, and on the left-hand side you will see Durham, and you might also notice the coast located on the Gulf of Maine, which is part of the Atlantic Ocean. And you will see Great Bay and Estuary, which is defined as the place where the river meets the sea. The boundaries of this ecosystem are a little hard to define, as the fresh water from the rivers meet the salty water from the ocean here in the estuary. All the water is flowing from the rivers into the bay at once, but this animation allows you to understand how the river flows. You can see it flowing from the Winnicott River, which travels about 9 miles through Northampton and Greenland and enters the bay here. The Lamprey River starts in the mountains in Northwood and travels nearly 50 miles through Lee, Durham, and then finally through Newmarket and enters the bay here. The Squamscott River travels 6 miles from Exeter through Newfields and Stratham and then enters the bay right here. Now that shows how the fresh water enters the bay. Now let's look at how the salty water comes in from the sea. You have probably seen the tide come in or out when you were at the ocean. You may have had to move your blanket to stay dry when the water has moved up the beach. When the tide is high, the ocean level rises and salt water flows into Portsmouth Harbor, the Piscataqua River, and Great Bay. The water level in the bay goes up for a while, and then it slowly goes back down, and then about 6 hours later there is a low tide and the bay is at its lowest point. This pattern continues with about 2 high tides and 2 low tides happening almost every day. As you can imagine, this ecosystem is quite complex and can be challenging to understand. In a minute, you will meet Anna, a graduate student who is studying the Great Bay Estuary in order to understand it better and to work with many other scientists to help to monitor and protect it. Before you meet her, I would like you to pause the video now and take a few minutes to think about any problems that occur in this estuary and what questions you might have if you were a scientist studying an estuary.
My name is Anna, and I'm a graduate student at the University of New Hampshire, and I work in the Water Quality Analysis Lab, where we study rivers, lakes, and estuaries, and how different things dissolved in the water change in concentration over time and over different spatial scales. And so I study specifically the water quality of Great Bay Estuary, which is behind me in this video. And I look at things dissolved in the water in the estuary and also in the rivers like the Lamprey, the Squamsquat, and the Winniket that drain into the estuary. And the specific water quality solutes that I look at include nitrogen, phosphorus, and dissolved oxygen. And nitrogen and phosphorus are really important nutrients for aquatic plants. They help they're required for aquatic plants to grow. And they also can influence the health of the ecosystem. And so when nitrogen or phosphorus are in excess in the estuary, or there's too much of it, it can cause something called an algal bloom, where little macroscopic algae basically grow and grow and grow across the top of the water, and they block out all the sunlight. And so sunlight can't penetrate through the water down to the native eelgrass that's submerged in the estuary. And as the algae die, they get broken down by microbes, and that process depletes oxygen in the estuary. And so that's the third thing that I look at, which is dissolved oxygen. And that's the same as the oxygen we breathe, but just dissolved in the water. And when dissolved oxygen levels go down because the algae are being broken down, that can be really bad for the fish because they need a certain amount of oxygen in order to breathe through their gills. And so those are the three things that I study specifically. Nitrogen is also really important to estuaries because there's another species that grows called seaweed. And seaweed will grow when there's lots of nitrogen, and it will actually grow on top of eelgrass and shade the light out from the eelgrass that way as well. And so part of my job as a scientist is to help monitor the amount of nitrogen and phosphorus that enters the estuary so that resource managers in the region can make decisions about how to protect the estuary better. And so that can include things like regulating the amount of nitrogen that comes out of a wastewater treatment plant, putting in more wetlands adjacent to the rivers and the estuary to promote the filtration of water before it enters the estuary. And so I study how nitrogen, phosphorus, and dissolved oxygen change over time, and also how they differ between the rivers and the estuary. And so I can add up all the nitrogen that enters the estuary from the rivers and subtract the amount that leaves the estuary and predict how much nitrogen stays within Great Bay Estuary. And then that allows us to ask really cool questions about how Great Bay processes that nitrogen. Or in other words, where does the nitrogen go once it's in the estuary? So for example, the nitrogen could go down and be buried in the sediments, or it could be used by the eelgrass, seaweed, and algae, or it could be denitrified, which means microbial communities take the nitrogen and use it in a special respiratory pathway and turn it into N2 gas, which is the most dominant gas in our atmosphere. Our atmosphere is made up of about 78% dinitrogen or N2 gas.
To protect the estuary, Anna and other scientists study how three things move in and out of this ecosystem. Do you remember what they are? She is measuring the amounts of nitrogen, phosphorus, and oxygen in the water and how these amounts change over time. You may not have heard of nitrogen and phosphorus before, but they are required for plants to grow. Nitrogen is in the air and can also drain into the bay through wastewater treatment plants, farmers' fields, and lawns that have been fertilized. If there is too much nitrogen, there might be too much plant growth, and that could have a bad effect on the ecosystem of the estuary because the seaweed and algae will grow on the top of the water and block out the sunlight, making it hard for the eelgrass to grow. If there is too little nitrogen, the plants won't be able to grow either. It is interesting to think that the oxygen we breathe in the air is also in the water. Just like the trees and plants on land produce oxygen through a process called photosynthesis, the underwater plants produce oxygen through this process as well. All fish and other animals require oxygen to survive. Now, how does Anna measure how much nitrogen, phosphorus, and oxygen is in the estuary? Where does it come from and how does it leave? You could pause the video now and talk with a partner about this, or draw a picture to show how everything is cycling into, within, and out of the ecosystem in the estuary.
So as a water quality scientist, part of my job involves collecting water samples so that we can analyze them for different nutrient concentrations and figure out how much nitrogen or how much phosphorus is in the water. And so in order to collect a water sample, I have three tools, important tools. The first is a syringe with the volume of water marked on it. So you start at 0 and you can collect all the way up to 60 milliliters. And you just, to draw water in, you just pull back on your syringe to fill it up. My next tool is a special filter because when we're collecting water samples, we don't want any sediment in our water sample. We only want the things that are dissolved in the water. And so we have to filter all the particles out of the water sample. And so in order to do that, I put one of these filters, and there's already one in mine, into a filter holder, and the water will be pushed through this in order to filter it. And the last tool I have is my sample bottle. And so this is my sample bottle, and this is where all my filtered water will go. And so I'm going to collect some water. So you can see I have water in my syringe, and then in order to make sure that there's no contamination from my syringe, what I'm going to do is I'm going to pull some air into my syringe and I'm going to shake it. And then I'm going to get rid of this water because what I'm doing is I'm rinsing my syringe. And then I'm going to get another sample of water. And now I'm going to put my filter on. I'm going to put some water through my filter. And then I'm going to rinse my sampling bottle with a little bit of filtered water. And then I'll get rid of that. And then I'm going to fill my sampling bottle. And so that's what a sampling bottle full of water would look like. And it's really important when you sample that you don't touch the inside of the bottle or the inside of the cap of the bottle, because your hands actually have some forms of nitrogen on them that can get into your sample and contaminate it. Or if it's a really hot sampling day and you're super sweaty, you can actually contaminate your sample with salt from your sweat. And that's something else that we can measure in our lab. And so you never want to touch the inside of a sampling bottle as a good scientist.
Once Anna has collected the water samples, she brings them back into her lab where she has tools and machines that help to measure the amount of nitrogen, phosphorus, and oxygen in the water. Once she gets this information, the work has really just begun. She needs to put her data into a form that makes the information easier to understand. She might make something called a graph, which makes her data look more like a picture, helping her to do the important step, which scientists call analyzing and interpreting data. Scientists don't just collect information, they also work to make sense of it. Anna will explain how she analyzes or makes sense of her data and how it helps her to understand what is happening in the Great Bay Estuary even more.
So I shared with you guys 2 graphs. One of them is discharge over time for 3 rivers that drain into Great Bay Estuary. And discharge is just a fancy word that means volume of water that moves past a certain point over time. And so an example would be liters of water that move past a certain point in a river in a given amount of time. And then that time is usually given in seconds or days. And so the graph I shared with you shows discharge for the lamprey, the squamsquat, and the winnicott over time. And one of the cool patterns that you might notice on the graph is a really big spike in discharge around 2010. And the reason for that big spike was we had a very heavy rainfall year, and there was a lot of flooding that resulted from those heavy rainstorms. And as that water fell on the landscape, it moved downhill across parking lots and across forest floors into the river, the lamprey, or any of the other ones. And so you see this really big peak in discharge as that water is accumulating throughout the watershed and enters the river. And the second graph I shared with you is dissolved oxygen concentrations in 3 rivers and within the Great Bay Estuary itself over time. And the really cool thing about that graph is you can compare your different sites to one another. And so you should see roughly 5 lines that are different colors that represent the different sites, but it's kind of hard to see them because they're all following the same pattern over time. And so that tells us that dissolved oxygen concentrations are very similar at all the sites. And part of the reason for that is because dissolved oxygen concentration in the water is driven by temperature. And so colder water can actually hold more dissolved oxygen than warmer water. And so that pattern or that oscillation that you see over time is seasonal because in the winter the water is a lot colder in Great Bay and in the rivers. And so dissolved oxygen concentrations go up because the water can hold more. And in the summer, the estuary gets warmer. It's great swimming weather, but there's less oxygen in the water because warm water can't hold as much. So estuaries are defined as where a river meets the sea. And they're very unique ecosystems because they represent the mixing of fresh water from a river and salty water from the ocean. And so they're very unique environments where different species of fish, crustaceans, and plants come together to create a unique environment. Part of what makes an estuary so important is it is often referred to as a coastal filter. And what I mean by that is if a river has a really big input of nitrogen, an estuary can actually filter some of that nitrogen out of the water before it goes out to the coastal ocean. And that can be through burying it in the sediments, being taken up by eelgrass, or being lost out to the air. And another reason estuaries are really important is because they provide habitat, specifically nursery habitat for juvenile fish and other species of organisms. And so they're where all the baby organisms come to live and stay. And so protecting estuaries is really important so that we can protect those important species. So the other thing I wanted to share with you guys today are 2 conceptual diagrams that I made. The first one looks at the hydrologic cycle of Great Bay Estuary. And the hydrologic cycle is basically the study of how water moves through an ecosystem. And so the blue arrows on the diagram show the different ways in which water can enter or leave the estuary. And so, for example, when it rains, precipitation can fall directly into the estuary, or that rain can fall onto the landscape and drain into wetlands or into a tributary or a river, and then the water, once it's in the river, can flow into the estuary. The other way that water enters an estuary is with the tides, and so water will come in with high tide and then it will leave during low tide, and that causes fluctuations in the level of water within the estuary. And studying the movement of water is really important for water quality management, because the movement of water is what carries all of those important things that are dissolved in the water. So the nitrogen, the phosphorus, the oxygen, all of those things are dissolved in river water or in precipitation, and they get transported from different parts of the world back into our ecosystem, and they end up in the estuary. And the other reason it's important is because the tidal cycle, the in and out of high and low tide, also will create an opportunity for dissolved things to move in and out of the estuary and flush out to the coastal ocean. And the second diagram that I made for you all shows the biogeochemistry of Great Bay. And biogeochemistry is a big word that basically looks at the biological, or the life-giving, the chemical, and the geological attributes of an ecosystem. So we look at the rocks, the chemical reactions, and the life, or the plants and the animals that interact in the estuary and move things like nitrogen around. And so the biogeochemical cycle that I showed you all shows some of the possible pathways or ways in which nitrogen can move in and out of the estuary. And so nitrogen that's dissolved in the river might come down river and enter the estuary, where it could then be taken up by a plant. And so that would be nitrate molecules being converted into organic nitrogen that's inside a plant or inside you and me. We also use nitrogen. That nitrogen could also get buried into the sediments. It could be denitrified, which is a process that happens when there's no oxygen, and it can go from nitrate to dinitrogen gas, or it could just be flushed from Great Bay out to the coastal ocean or the Gulf of Maine. And so those are just a few examples of biogeochemical cycles that happen in Great Bay Estuary. There are a lot more that are studied by a lot of different scientists, but I'm just working on that one small piece of a bigger ecosystem.